Basics of Medical Biochemistry. First edition, 2022. Chapter 10. Metabolism of Carbohydrates, Part 1 of 3. By Professor Dr. Muhammad Jawad. MBBS, Tao, M.Phil Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 10. Metabolism of Carbohydrates. 10.1 Introduction. Carbohydrates are central to many essential metabolic pathways. Carbohydrate metabolism is the whole of the biochemical processes responsible for the metabolic formation, breakdown, and interconversion of carbohydrates in living organisms. It is a fundamental biochemical process that ensures a constant supply of energy to living cells. Carbohydrate metabolism starts in the mouth, where the enzyme salivary amylase begins to break down complex sugars into monosaccharides. These can then be transported across the intestinal membrane into the bloodstream and then to body tissues. The most important carbohydrate is glucose. The stored form of glucose is called glycogen and is primarily found in the liver and muscle. Glucose is metabolized to carbon dioxide, water, and ADP in the cells by three consecutive processes: glycolysis, tricarboxylic acid, TCA, cycle, and electron transport chain. Other important pathways in carbohydrate metabolism include the pentose phosphate pathway or hexose monophosphate, HMP, shunt, glycogenesis, glycogenolysis, hexose interconversions, gluconeogenesis, oxidative phosphorylation, and Cori cycle. Figure 10.1 Outline of carbohydrate metabolism showing relationship with lipid and protein metabolism, HMP, hexose monophosphate, TCA, tricarboxylic acid. Source: Harper's Illustrated Review Biochemistry 31st edition. 10.2 Glycolysis. Glycolysis is a catabolism of glucose. It is a cascade of enzymatically controlled reactions that convert glucose into 2-pyruvates, in aerobic condition, or lactate molecules, in anaerobic condition, and produces ADP along with NADH plus H+. It occurs intracellularly in the cytoplasm of cells of almost all tissues of the body. Glycolysis is also known as the Embden-Meyerhof pathway. It is especially important in some tissues like RBCs, absence of mitochondria, depend on glycolysis as the main source of energy. Contracting muscles due to the occlusion of blood vessels by muscular contraction decreases oxygen supply. The cornea, lens and some parts of the retina have a limited blood supply and lack mitochondria. Table 10.1 Types of glycolysis. Aerobic glycolysis. Anaerobic glycolysis occurs in the presence of oxygen. Produce pyruvic acid. Produce comparatively more energy. 6 moles of ADP per mole of glucose. Occurs in the absence of oxygen. Produce lactic acid. Produce comparatively less energy. 2 moles of ADP per mole of glucose. 10.2.1 Steps of glycolysis. Following are the reactions of glycolysis. 1. Conversion of glucose to glucose 6-phosphate. Glucose is converted to glucose 6-phosphate by phosphorylation at the cost of ADP. This reaction is catalyzed by hexokinase in all extrahepatic tissues. There are 4 isozymes, I4, of hexokinase, found in most tissues of the body. In the liver and beta cells of the endocrine pancreas, this reaction is carried out by glucokinase, hexokinase 4. The reaction is endergonic in nature and enzymes need Mg2+ ions as a cofactor. Reversal needs glucose 6-phosphate phosphatase, or simply glucose 6-phosphatase, found only in the liver. By phosphorylation, glucose is trapped within the cell and postprandial hyperglycemia is prevented. Glucose plus ADP. Glucose 6-phosphate plus ADP. Glucokinase in beta cells serves as a glucose sensor, amplifying insulin secretion as blood glucose rises. In pancreatic beta cells, glucokinase is a key regulator enzyme. In the presence of fructose 6-phosphate, the glucokinase regulatory protein, GKRP, binds to and inhibits liver glucokinase. Both phosphate esters act by binding to the regulatory protein, and by presumably causing changes in its conformation. The regulatory protein behaves as a fully competitive inhibitor of glucose. The advantage for the liver to have both hexokinase and glucokinase. Glucokinase enables the liver to remove glucose from the blood when hexokinase is saturated, ensuring that glucose is captured for later use. In the liver, glucokinase plays a key role in the ability to store glucose as glycogen, particularly in the postprandial state. Table 10.2 Difference between hexokinase and glucokinase. Hexokinase. Glucokinase. Found in all tissues, except the liver and beta cells of the endocrine pancreas. Affinity for glucose. Low Km. Can act on all hexoses. Stimulated by hypoglycemia. Inhibited by glucose 6-phosphate. Present only in liver parenchymal and beta cells of the endocrine pancreas. Affinity for glucose. High Km. Acts only on glucose. Stimulated by hyperglycemia. Not inhibited by glucose 6-phosphate. 2. Conversion of glucose 6-phosphate to fructose 6-phosphate. It is the reversible isomerization of glucose 6-phosphate to fructose 6-phosphate by the enzyme phosphohexose isomerase or phosphoglucose isomerase. As a result of this isomerization, aldose is changed to ketose. Glucose 6-phosphate fructose 6-phosphate. 3. Conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1, PFK1. This enzyme is activated by AMP, ADP, fructose 6-phosphate, and fructose 2,6-bisphosphate. The reaction is endergonic and reversal needs fructose 1,6-bisphosphate phosphatase. PFK1 is an allosteric enzyme and acts as a major regulatory enzyme of glycolysis. Therefore, the rate-limiting step in glycolysis is the reaction catalyzed by PFK1. Fructose 6-phosphate plus ADP fructose 1,6-bisphosphate plus ADP. Fructose 2,6-bisphosphate, the most potent activator of PFK1, is formed from fructose 6-phosphate by phosphofructokinase 2, PFK2. 4. Cleavage of fructose 1,6-bisphosphate to 2 molecules of triose phosphate, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The molecule of fructose 1,6-bisphosphate is cleaved at carbon atoms number 3 and 4 to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme fructose 1,6-bisphosphate aldolase. Fructose 1,6-bisphosphate glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. There is aldolase B, which can also cleave fructose 1,6-bisphosphate as well as performs a role in the metabolism of dietary fructose. 5. Interconversion of 2 triose phosphates. The 2 triose phosphate molecules so produced are easily interconvertible by the enzyme triose phosphate isomerase. However, in glycolysis dihydroxyacetone phosphate is rapidly converted to glyceraldehyde 3-phosphate. In this way 2 molecules of glyceraldehyde 3-phosphate are produced from 1 molecule of fructose 1,6-bisphosphate. Now subsequent reactions of glycolysis i.e., from glyceraldehyde 3-phosphate onward will be considered a duplicate. Dihydroxyacetone phosphate glyceraldehyde 3-phosphate. 6. Conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. During this redox reaction, H+ ions are accepted by NAD+ and it is reduced to NADH plus H+. This reducing equivalent is carried to mitochondria by glyceraldehyde 3-phosphate or malate aspartate shuttle, where it is oxidized by oxidative phosphorylation to yield ADP. It is the first reaction of glycolysis which yield energy. NAD plus NADH plus H plus. Glyceraldehyde 3-phosphate plus Pi1,3-bisphosphoglycerate. 7. Conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. This step is catalyzed by the enzyme phosphoglycerate kinase. 1-ADP is also formed during the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. This step is an example of substrate-level phosphorylation. The reaction is reversible by the same enzyme. 1,3-bisphosphoglycerate plus ADP 3-phosphoglycerate plus ADP. 8. Conversion of 3-phosphoglycerate to 2-phosphoglycerate. Phosphoglycerate mutase converts 3-phosphoglycerate to 2-phosphoglycerate. 3-phosphoglycerate 2-phosphoglycerate. 9. Conversion of 2-phosphoglycerate to phosphenol pyruvate. PEP. The 2-phosphoglycerate is dehydrated to phosphenol pyruvate by the enzyme enolase, in the presence of Mg2+ or Mn2+ ions. Enolase is inhibited by fluoride ions, that's why the addition of fluoride to mouthwashes and toothpaste is an established strategy to prevent dental caries by resisting glycolysis. Similarly, blood samples should be fluorinated to stop glycolysis in RBCs to measure blood glucose levels accurately. 2-phosphoglycerate phosphenol pyruvate plus H2O. 10. Conversion of phosphenol pyruvate to pyruvate. It is another reaction of substrate-level phosphorylation in glycolysis, producing 1-ADP. It is catalyzed by the enzyme pyruvate kinase in the presence of K+ or either Mg2+ or Mn2+ ions. Pyruvate kinase is an allosteric enzyme inhibited by ADP, alanine, fatty acid, and acetyl-CoA. It is the only irreversible reaction of glycolysis. Phosphenol pyruvate plus ADP pyruvate plus ADP. Net reactions of glycolysis. C6H12O6 plus 2 NAD plus 2 ADP plus 2 Pi2CH3 CoQ plus 2 NADH plus H plus 2 ADP plus 2 H2O. Glucose, pyruvate. Figure 10.2 Reactions of glycolysis. 10.2.2 Energy yield of glycolysis, under anaerobic conditions equals 2 ADP for the anaerobic oxidation of 1 mole of glucose into 2 moles of lactate. Under aerobic conditions equals 2 ADP plus 2 NADH plus H plus, 1 NADH plus H plus equals 2.5 ADP, provided that NADH is transported to mitochondria by malate shuttle. 10.2.3 Interconversion of pyruvate and lactate, under anaerobic conditions, i.e., absence of O2 or mitochondria, NADH plus H plus is reoxidized to allow glycolysis to continue by lactate dehydrogenase, LDH, that reversibly reduces pyruvate into lactate. This reaction is readily reversible. Figure 10.3 Lactate dehydrogenase reaction, interconversion of pyruvate and lactate. 10.2.4 Fate of pyruvate. It gives glucose by reversal of glycolysis reactions, gluconeogenesis. It gives lactate in the anaerobic condition which is gluconeogenic. It gives oxalostate. It gives active acetate, acetyl-CoA, that links carbohydrates, lipids, and proteins that oxidized in the TCA cycle. It gives alanine by transamination. Pyruvate passes into the mitochondria by a special transporter on the inner mitochondrial membrane. 10.2.5 Regulation/control of glycolysis. It is regulated by enzymes, hormones as well as in vitro by certain chemical agents. 1 Key regulatory enzymes, hexokinase, accumulation of glucose 6-phosphate that allosterically inhibits hexokinase. Phosphofructokinase 1, PFK1, is stimulated by high levels of fructose 6-phosphate, fructose 2,6-bisphosphate, in the liver, ADP, AMP, and Pi. It is inhibited by ADP, low pH, and citrate. Pyruvate kinase is inhibited also by excess ADP, alanine, and acetyl-CoA and stimulated by fructose 1,6-bisphosphate, ADP and AMP. 2 Hormonal regulation, insulin, stimulates glycolysis. It also induces glucose transporters to provide cells with glucose for glycolysis. Adrenaline and glucagon are inhibitors of glycolysis by inhibiting pyruvate kinase. 3. In vitro inhibition of glycolysis. The glycolysis can be inhibited by 2-deoxyglucose, which inhibits hexokinase and glucokinase. Iodoacetate inhibits glyceraldehyde 3-phosphate dehydrogenase. Fluoride inhibits enolase. Hence, for accurate measurement of blood glucose levels, the samples should be fluorinated to stop glycolysis in RBCs. Arsenate competes with phosphate and interferes with the formation of ADP by inhibiting the production of 1,3-bisphosphoglycerate. 10.2.6 Biomedical importance of glycolysis. It is the major source of energy in certain tissues, example RBCs and skeletal muscles. It provides the pyruvic acid needed for the TCA cycle. It is a link with fat metabolism, example dihydroxyacetone phosphate can be changed into glycerol 3-phosphate in adipose tissue. It has a link with amino acid metabolism, example 3-phosphoglycerate can be changed into serine and pyruvate into alanine and vice versa. It provides various intermediates for other metabolic pathways. It is the major source of lactic acid which is gluconeogenic. 10.2.7 Rappaport-Lubering cycle or Rappaport-Lubering shunt. It is regarded as a shunt pathway of glycolysis to dissipate or waste the energy not needed by RBCs. In mature RBCs, the first site for ADP generation in glycolysis may be bypassed. Bisphosphoglycerate mutase catalyzes the conversion of 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate, which is converted to 3-phosphoglycerate by 2,3-bisphosphoglycerate phosphatase. Figure 10.4 Formation of 2,3-bisphosphoglycerate in RBCs. 10.2.8 Clinical significance of glycolysis. It is regarded as a primary source of energy in carbohydrate metabolism. It can produce energy in skeletal muscles even in absence of oxygen. Heart muscles have poor glycolytic activity and poor survival under ischemic conditions. An increased rate of glycolysis is observed in cancer cells with resultant lactic acidosis. Congenital deficiencies of enzymes like hexokinase and pyruvate kinase can produce hemolytic anemia. 10.2.9 Diseases associated with impaired glycolysis. 1. Lactic acidosis. This depletes the blood alkali reserve leading to hyperlactemia and lactic acidosis when blood lactate exceeds its normal level. Uncontrolled lactic acidosis could cause coma. Causes of hyperlactemia include: severe exhaustive muscle exercise increases lactate production. Anoxia as in shock and diseases that impair blood flow, respiratory disorders and severe anemia. Liver diseases decrease lactate utilization and extensive cancer increases it due to its anaerobic glycolytic activity. 2. Pyruvate kinase deficiency, hemolytic anemia due to genetic reduction of pyruvate kinase activity to 5%-25% of the normal level. This causes a decreased rate of glycolysis and leads to a deficiency of ADP production in RBCs. RBCs cannot control their size, and swell and lysis occur, hemolysis, due to the stoppage of the ADP-dependent membrane pumps. 3. Hexokinase deficiency. Genetic deficiency of hexokinase causes a decreased rate of glycolysis and low 2,3-bisphosphoglycerate level leading to high hemoglobin affinity to O2 that will be less available for tissues. 4. Glucokinase deficiency. Maturity onset diabetes of young type 2, MODY2, is a rare form of diabetes of the young characterized by impaired insulin secretion and hyperglycemia. MODY2 is due to several mutations in the GCK gene on chromosome 7 for glucokinase. Glucokinase serves as the glucose sensor for the pancreatic beta cell. Normal glucokinase triggers insulin secretion as the glucose exceeds about 90 mg/dl or 5 mol/L. Table 10.3 Prominent features of glycolysis. Reaction No. Substrates. Products. Enzyme. Nature of reaction. Remarks. 1. Glucose plus ADP. Glucose 6-phosphate plus ADP. Hexokinase/glucokinase. Phosphorylation. The reaction is endergonic in nature and reversal needs glucose 6-phosphate phosphatase. 2. Glucose 6-phosphate. Fructose 6-phosphate. Phosphohexose isomerase. Isomerization. The reaction is reversible. 3. Fructose 6-phosphate plus ADP. Fructose 1,6-bisphosphate plus ADP. Phosphofructokinase 1. Phosphorylation. The reaction is endergonic in nature and reversal needs fructose 1,6-bisphosphate phosphatase. 4. Fructose 1,6-bisphosphate. Glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. Fructose 1,6-bisphosphate aldolase. Cleavage of bisphosphate into 2-triose phosphate. Reaction is reversible. 5. Dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate. Triose phosphate isomerase. Isomerization. Subsequent reactions of glycolysis will be in duplicate. 6. Glyceraldehyde 3-phosphate plus pi plus NAD plus 1,3-bisphosphoglycerate plus NADH plus H plus. Glyceraldehyde 3-phosphate dehydrogenase. Oxidation, dehydrogenation. 1 NADH plus H plus produced, equivalent of 2.5 ADP, star. 7. 1,3-bisphosphoglycerate plus ADP. 3-phosphoglycerate plus ADP. Phosphoglycerate kinase. Substrate level phosphorylation. Formation of 1 ADP. 8. 3-phosphoglycerate. 2-phosphoglycerate. Phosphoglycerate mutase. Intramolecular change. Shifting of phosphate from C3 to C2. 9. 2-phosphoglycerate. Phosphenol pyruvate, PEP, plus H2O. Enolase. Dehydration. PEP is a high-energy compound. Enolase can be inhibited by fluoride. 10. PEP plus ADP. Pyruvate plus ADP. Pyruvate kinase. Substrate level phosphorylation. Formation of 1 ADP. Asterisk provided that NADH is transported to mitochondria by malate shuttle. 10.3 Cori cycle. The Cori cycle or lactic acid cycle is named after its discoverers, Carl Cori and Gerty Cori, who jointly won the 1947 Nobel Prize in Biochemistry for their discovery of the Cori cycle. It refers to the metabolic pathway in which lactate produced by anaerobic glycolysis in the muscles moves to the liver and is converted to glucose, which then returns to the muscles and is converted back to lactate. The muscular activity requires energy, which is provided by the breakdown of glycogen in the skeletal muscles. The breakdown of glycogen, a process known as glycogenolysis, releases glucose in the form of glucose 6-phosphate. The released glucose 6-phosphate is readily fed into glycolysis, a process that provides ADP to the muscle cells as an energy source. During the muscular activity, the store of ADP needs to be constantly replenished. When the supply of oxygen is sufficient, this energy comes from feeding pyruvate, one product of glycolysis, into the Krebs cycle. Instead of accumulating inside the muscle cells, lactate produced by anaerobic fermentation is taken up by the liver. This initiates the other half of the Cori cycle. In the liver, gluconeogenesis occurs. The glucose is then supplied to the muscles through the bloodstream. It is ready to be fed into further glycolysis reactions. If muscle activity has stopped, the glucose is used to replenish the supplies of glycogen through glycogenesis. Overall, the glycolysis part of the cycle produces 2 ADP molecules at 6 ADP molecules consumed in the gluconeogenesis part. Each turn of the cycle must be maintained by a net consumption of 4 ADP molecules. As a result, the cycle cannot be sustained indefinitely. The intensive consumption of ADP molecules indicates that the Cori cycle shifts the metabolic burden from the muscles to the liver. Significance of the Cori cycle. The cycle's importance is based on the prevention of lactic acidosis in the muscle under anaerobic conditions. However, normally before this happens the lactic acid is moved out of the muscles and transported to the liver. The cycle is also important in producing ADP, an energy source, during muscle activity. The Cori cycle functions more efficiently when muscle activity has ceased. This allows the oxygen debt to be repaid such that the TCA cycle and electron transport chain can produce energy at peak efficiency. Figure 10.5 Cori cycle. 10.4 Formation of acetyl-CoA from pyruvate. Pyruvate is formed in the cytosol as a product of glycolysis. For entry into the TCA cycle, it has to be converted to acetyl-CoA. Oxidative decarboxylation of pyruvate to acetyl-CoA is catalyzed by the pyruvate dehydrogenase (PDH) complex in the mitochondria. Figure 10.6 Formation of acetyl-CoA from pyruvate. The PDH complex consist of three enzymes: pyruvate dehydrogenase or pyruvate decarboxylase (E1), dihydrolipoyl transacetylase (E2), and dihydrolipoyl dehydrogenase (E3). It has 5 cofactors: CoA, NAD+, lipoic acid or lipomide, thiamine pyrophosphate, TPP, and fat. CoA and NAD+ participate stoichiometrically in the reaction. The other 3 cofactors have a catalytic function. TPP is the cofactor for E1, lipoic acid and CoA are the cofactors for E2 while fat and NAD+ are the cofactors of E3. PDH complex also has 2 regulatory enzymes named pyruvate dehydrogenase kinase, PDH kinase, and pyruvate dehydrogenase phosphatase, PDH phosphatase. The PDH complex reaction is irreversible. 1 carbon of pyruvate is oxidized to CO2 and 1 NADH plus H plus is formed by the PDH reaction. This is called as activation of pyruvate. In the TCA cycle, intermediates are activated by the formation of high-energy thioester bonds. Enzymes of the PDH complex and the TCA cycle, except succinate dehydrogenase, are found in the mitochondrial matrix. Pyruvate translocase is an antiporter present in the inner mitochondrial membrane that allows entry of a molecule of pyruvate in exchange for a hydroxide ion. Although the formation of acetyl-CoA from pyruvate takes place in mitochondria, but it is not the part of TCA cycle. Figure 10.7 Reactions of the pyruvate dehydrogenase complex, TPP, thiamine pyrophosphate. Regulation of PDH complex. The pyruvate dehydrogenase complex may be allosterically activated by fructose 1,6-bisphosphate and is inhibited by NADH and acetyl-CoA. It is regulated by covalent modification through the action of a specific kinase and phosphatase. The kinase and phosphatase are regulated by changes in NADH, acetyl-CoA, pyruvate, and insulin. PDH kinase is stimulated by ADP, NADH, and acetyl-CoA. It is inhibited by pyruvate. Insulin, NAD+, and ADP stimulate PDH phosphatase. Figure 10.8 Regulation of pyruvate dehydrogenase complex, i.e., PDC, PDPs, pyruvate dehydrogenase phosphatase, PDKs, pyruvate dehydrogenase kinase. Clinical aspects. Pyruvate dehydrogenase complex deficiency is one of the most common neurodegenerative disorders associated with abnormal mitochondrial metabolism. A malfunction of the TCA cycle deprives the energy of the body. PDH complex deficiency is caused by genetic changes, mutations or pathogenic variants, in any of the genes that are responsible to make the enzymes of the pyruvate dehydrogenase complex. Leigh syndrome, subacute necrotizing encephalomyelopathy. It is caused by defects in mitochondrial ADP formation due to PDH complex deficiency. Its symptoms include developmental delay, intellectual disability, mixed acid-base disturbance with mild to moderate elevations in lactic acid and ketone bodies in the blood, lactic acidosis/ketoacidosis, abdominal pain, vomiting, tiredness and muscle weakness. A medicine, dichloroacetate, may be effective in some patients. Diet high in fats and low in carbohydrates, ketogenic diet, is also recommended. This can help to prevent lactic acidosis but bring about no improvement in neurological symptoms. Wernicke-Korsakoff, an encephalopathy psychosis syndrome. It occurs due to thiamine deficiency, and may be seen in alcohol abusers. In this condition, brain cells are unable to furnish adequate amounts of ADP, because one of the cofactors of the PDH complex, i.e., thiamine, is deficient. 10.5 Tricarboxylic acid, TCA, cycle. It is also known as the citric acid cycle or Krebs cycle after the name of scientist Hans Adolf Krebs, who described it in 1937. It is a cyclic pathway, and all of its intermediates are derivatives of citric acid, a tricarboxylic acid. It is regarded as the next step in intracellular glucose metabolism. It takes place in the mitochondria and is initiated by acetyl coenzyme A, acetyl-CoA, which is an oxidized derivative of the pyruvate produced during glycolysis. Acetyl-CoA, a 2-carbon molecule, starts the cycle by condensation with the 4-carbon oxalostate. Each of the 2 carbons of acetyl-CoA is oxidized and removed as CO2 in 2 separate reactions. Oxalostate is regenerated. The TCA cycle requires oxygen and releases carbon dioxide and water as a byproduct. It is composed of a series of redox reactions that generate the high-energy molecules NADH, FADH2, and ADP. For each glucose molecule, 2 pyruvates are produced via glycolysis. Thus, the citric acid cycle goes around twice and produces 2 carbon dioxide molecules, 3 NADH, 1 FADH2, and 1 ADP for each turn. While the citric acid cycle does not itself produce much ADP, the major energy currency of the cell, the NADH and FADH2 molecules, act as electron carriers that shuttle into the electron transport chain for oxidative phosphorylation and high-energy production. 10.5.1 Steps of TCA cycle. There is a total of 8 steps in the TCA cycle, all of them except 3 are reversible. 1. Formation of citrate by condensation of oxalostate and acetyl-CoA. In step 1 of the TCA cycle, the 2-carbon compound, acetyl-CoA, participates in a condensation reaction with the 4-carbon compound, oxalostate, to produce citrate by citrate synthase. The reaction is irreversible. Acetyl-CoA plus oxalostate plus H2O citrate plus CoA. 2. Isomerization of citrate to isocitrate. It involves moving the hydroxyl group in the citrate molecule by a sequential dehydration and hydration reaction, to form the isocitrate isomer with cis-aconitate as the intermediate. A single enzyme, aconitase, aconitate hydratase, performs this 2-step process. 2A citrate rightward arrow cis-aconitate plus H2O. 2B cis-aconitate plus H2O rightward arrow isocitrate. The net reaction is citrate isocitrate. 3. Formation of alpha-ketoglutarate by oxidative decarboxylation of isocitrate. It is the first oxidative decarboxylation step of the TCA cycle. The reaction is catalyzed by the enzyme isocitrate dehydrogenase. Isocitrate is converted to alpha-ketoglutarate. The reaction involves 3A dehydrogenation of isocitrate to oxalosuccinate. 3B oxalosuccinate is an unstable intermediate that is spontaneously decarboxylated to give alpha-ketoglutarate with the liberation of CO2. In addition to decarboxylation, this step produces a reduced nicotinamide adenine dinucleotide, NADH, cofactor. The reaction is irreversible. Isocitrate plus NAD plus alpha-ketoglutarate plus NADH plus H plus CO2. 4. Formation of succinyl-CoA by oxidative decarboxylation of alpha-ketoglutarate. This step is performed by a multi-enzyme complex, the alpha-ketoglutarate dehydrogenase complex. It is composed of 3 enzymes and 5 coenzymes like the pyruvate dehydrogenase complex. The reaction is irreversible. Alpha-ketoglutarate plus CoA plus NAD plus succinyl-CoA plus NADH plus H plus CO2. Succinyl-CoA is also produced from propionyl-CoA, derived from the metabolism of an odd number of carbon atoms containing fatty acids. It can be used in heme synthesis or converted to pyruvate for gluconeogenesis. 5. Formation of succinate. succinyl-CoA is a high-potential energy molecule. It has a high-energy thioester bond. The energy stored in this molecule is used to form a high-energy phosphate bond in a guanine nucleotide diphosphate, GDP, molecule. The reaction is reversible and catalyzed by the enzyme succinyl-CoA synthetase, succinate thiokinase. This reaction is an example of substrate-level phosphorylation. Succinyl-CoA plus GDP plus pi-succinate plus GTP plus CoA. In animals, GDP is the substrate in the reaction with the formation of GTP, which is then used to form ADP by nucleoside diphosphate kinase. In plants and bacteria, ADP is formed in the succinyl-CoA synthetase catalyzed reaction by phosphorylation of ADP directly. 6. Formation of fumarate by oxidation of succinate. It is a flavin-dependent dehydrogenation reaction. In this step, succinate is converted to fumarate by succinate dehydrogenase. In this reaction, a C-C bond is oxidized to produce a C=C bond. This oxidation is energetically costlier than oxidizing a CO bond. The redox coenzyme for this reaction is Fad, and not NAD+. Fad is a more powerful oxidizing agent compared to NAD+. Fad is covalently bound to the succinate dehydrogenase molecule. Succinate plus Fad fumarate plus FADH2. 7. Formation of malate by hydration of fumarate. In this step, fumarate undergoes stereospecific hydration of the C=C double bond, catalyzed by the enzyme fumarase or fumarate hydratase to produce malate. The reaction is freely reversible. Fumarate plus H2O malate. 8. Formation, regeneration, of oxalostate. Malate is dehydrogenated to produce oxalostate by the enzyme malate dehydrogenase. This is a highly endergonic reaction, and so, the equilibrium strongly favors the reactants over the products. This step also produces a reduced nicotinamide adenine dinucleotide, NADH, cofactor. L-malate plus NAD plus oxalostate plus NADH plus H+. Oxalostate reacts with the new acetyl-CoA molecule to repeat the TCA cycle. The overall reaction of the TCA cycle. Acetyl-CoA plus 3 NAD plus Fad plus GDP plus pi plus 2H2O2 Co2 plus CoA plus 3 NADH plus H plus FADH2 plus GTP. Summary of the TCA cycle in terms of ADP, energy gain. Total NADH plus H plus in one TCA cycle equals 3 molecules. 3 NADH plus H plus molecules would yield 3 times 2.5 equals 7.5 ADP molecules in electron transport system. Total FADH2 in one TCA cycle equals 1 molecule. 1 FADH2 molecule would yield 1 times 1.5 equals 1.5 ADP in electron transport system. ADP production at substrate level equals 1. Total ADP generation by one TCA cycle equals 7.5 plus 1.5 plus 1 equals 10. Figure 10.9 Reactions of TCA cycle. 10.5.2 Regulation of the TCA cycle. The regulation of the TCA cycle is largely determined by product inhibition and substrate availability. It is also controlled by the regulation of several enzyme activities such as citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase complex. These enzymes catalyze reactions No. 3 and 4 of the TCA cycle. All these 3 reactions are irreversible. Citrate synthase is activated by ADP and pi while inhibited by ADP and citrate. Isocitrate dehydrogenase is activated by ADP and Ca2+ but inhibited by NADH plus H plus and ADP. Alpha-ketoglutarate dehydrogenase complex is activated by ADP and Ca2+ but inhibited by succinyl-CoA, NADH plus H plus and ADP. TCA cycle is regulated by the ratio of ADP, pi/ADP. The demand for a new synthesis of ADP is limited under resting conditions, with a high cell energy charge and, although the proton motive force, PMF, is high, the flow of protons back into the mitochondria through ADP synthetase is minimal. When energy demands are increased, such as during exertion and severe physical activity, cytosolic ADP rises and is exchanged with intramitochondrial ADP via the transmembrane adenine nucleotide carrier ADP/ADP translocase. Increased intramitochondrial concentrations of ADP cause the PMF to become discharged as protons pour through ADP synthase, regenerating the ADP pool. Overall, in the case of elevated ADP, the TCA cycle is accelerated to generate ADP. Inhibitors of the TCA cycle. Fluorostate inhibits aconitase, it is a non-competitive inhibitor. Arsenate, a pentavalent form of arsenic, or arsenite, a trivalent form of arsenic, can inhibit PDH complex and alpha-ketoglutarate dehydrogenase complex. These are also non-competitive inhibitors. Malonate inhibits succinate dehydrogenase by competitive inhibition. 10.5.3 Biomedical importance of the TCA cycle. Energy production, 10 ADP per cycle. Acts as a final common pathway for the oxidation of all major nutrients to acetyl-CoA. It is a major source of succinyl-CoA which is used for the synthesis of hemoglobin and other porphyrins. It provides intermediates for the synthesis of non-essential amino acids. For example, many amino acids are synthesized starting with the transamination of alpha-ketoglutarate or oxalostate. Oxalostate is also the precursor of purines and pyrimidines via aspartate. Fatty acids and sterols are synthesized from citrate. It is an amphibolic pathway for gluconeogenesis, transamination, deamination, and lipogenesis, particularly in the liver. 10.5.4 Anaplerosis or anaplerotic reactions of the TCA cycle. Since the TCA cycle intermediates are used for anabolism, their concentration varies according to the needs of the cell. The reactions concerned to replenish or filling up the intermediates of the citric acid cycle are called anaplerotic reactions or anaplerosis. Following are the important anaplerotic reactions for the TCA cycle. Pyruvate carboxylase catalyzes the conversion of pyruvate to oxalostate. This is an ADP-dependent carboxylation reaction. Pyruvate plus CO2 plus ADP oxalostate plus ADP plus Pi. Pyruvate is converted to malate by NADP plus dependent malate dehydrogenase, malic enzyme. Pyruvate plus CO2 plus NADP plus H plus L malate plus NADP plus. Alpha-ketoglutarate can also be synthesized from glutamate by glutamate dehydrogenase. Figure 10.10 Role of TCA cycle as an amphibolic pathway. 10.5.5 Role of the citric acid cycle as an amphibolic pathway. The citric acid cycle is amphibolic in nature, i.e., serving in both catabolism and anabolism. The intermediates of the cycle can be drawn off and used as the starting material for a variety of biosynthetic products. At the same time, depleted cycle intermediates are replenished by various anaplerotic reactions. Table 10.4 Prominent features of the TCA cycle. Reaction No. Substrates. Products. Enzyme. Nature of reaction. Remarks. 1. Acetyl-CoA plus oxalostate plus H2O. Citrate plus CoA. Citrate synthase. Irreversible aldol condensation. Rate-limiting step. Increases the 4C oxalostate to a 6C citrate. The reaction is irreversible. 2. Citrate. Isocitrate. Aconitase. Dehydration and rehydration. Reversible isomerization. 3. Isocitrate plus NAD plus alpha-ketoglutarate plus CO2 plus NADH plus H plus isocitrate dehydrogenase. Oxidative decarboxylation. 1 NADH plus H plus produced, equivalent of 2.5 ADP. The reaction is irreversible. 4. Alpha-ketoglutarate plus NAD plus plus CoA. Succinyl-CoA plus NADH plus H plus plus CO2. Alpha-ketoglutarate dehydrogenase complex. Oxidative. Decarboxylation. 1 NADH plus H plus produced, equivalent of 2.5 ADP. Reaction is irreversible. 5. Succinyl CoA plus GDP plus pi. Succinate plus CoA plus GTP. Succinyl CoA synthetase, succinate thiokinase. Substrate level phosphorylation. 1. ADP produced via duct GTP by nucleoside diphosphate kinase. 6. Succinate plus FAD. Fumarate plus FADH2. Succinate dehydrogenase. Oxidation, dehydrogenation. 1 FADH2 produced. Equivalent of 1.5 ADP. 7. Fumarate plus H2O. L-malate. Fumarase, fumarate hydratase. Hydration. Hydration of the C equals C double bond of fumarate. 8. L-malate plus NAD plus oxalostate plus NADH plus H plus. Malate dehydrogenase. Oxidation, dehydrogenation. 1 NADH plus H plus produced, equivalent of 2.5 ADP. Regeneration of oxalostate. 10.5.6 ADP production from the complete oxidation of one glucose molecule. A total of 32 ATPs are gained by the complete oxidation of glucose. During glycolysis, a total of 4 ATPs are formed, out of which 2 are consumed. 2 NADH plus H plus are also produced during glycolysis, and each can furnish 2.5 ATPs via oxidation through the electron transport chain, etc., provided that they are transported to mitochondria by the malate shuttle. During the conversion of pyruvate to acetyl CoA, 2 molecules of NADH plus H plus are produced per glucose, which will yield 5 ATPs in the etc. The TCA cycle produces 2 molecules of ATPs for every molecule of glucose. The TCA cycle also produces 6 molecules of NADH plus H plus and 2 molecules of FADH2 per molecule of glucose. As each molecule of NADH plus H plus is equivalent to 2.5 ADP and each FADH2 is equal to 1.5 ATPs. No of ADP produced per molecule of glucose during glycolysis equals 4. No of ADP produced per molecule of glucose during glycolysis via NADH plus H plus equals 2 times 2.5 equals 5. No of ADP consumed per molecule of glucose during glycolysis equals -2. Net ADP produced per molecule of glucose during glycolysis equals 7. No of ADP produced per molecule of glucose during conversion of pyruvate to acetyl CoA equals 5. No of ADP produced per molecule of glucose by TCA cycle equals 20. Net ADP produced per molecule of glucose by complete oxidation, in aerobic condition, equals 32. Table 10.5 ADP formation by oxidation of glucose. Metabolic pathway. Reaction. No. Enzyme involved. Mode of reaction. ADP produced per mole of glucose. Glycolysis. 1. Hexokinase/glucokinase. Phosphorylation. 1. 3. Phosphofructokinase 1. Phosphorylation. 1. 6. Glyceraldehyde 3-phosphate dehydrogenase. Oxidation of 2 NADH and etc. 5 *. 7. Phosphoglycerate kinase. Substrate level phosphorylation. 2. 10. Pyruvate kinase. Substrate level phosphorylation. 2. Conversion of pyruvate to acetyl CoA. Pyruvate dehydrogenase. Complex. Oxidation of 2 NADH. In etc. 5. TCA cycle. 3. Isocitrate dehydrogenase. Oxidation of 2-NADH. In etc. 5. 4. Alpha-ketoglutarate dehydrogenase complex. Oxidation of 2-NADH. In etc. 5. 5. Succinyl-CoA synthetase. Succinate thiokinase. ADP formation via GTP. Binucleoside. Diphosphate kinase. 2. 6. Succinate dehydrogenase. Oxidation of 2-FADH2. In etc. 3. 8. Malate dehydrogenase. Oxidation of 2-NADH. In etc. 5. Net ADP produced per mole of glucose by complete oxidation, in aerobic conditions. 32. Asterisk provided that NADH is transported to mitochondria by malate shuttle. Exercise questions. MCQ 1. An athlete had just finished a 450 meters run. Which of the following metabolic product is expected to be raised in his muscle cells? Citrate, glycerol, lactate, oxalostate, pyruvate. MCQ 2. The phosphoglycerate kinase enzyme is blocked during an experimental study on the mechanism of energy production in a cell. Which of the following mechanisms related to carbohydrate metabolism about ADP production is restricted as a result of blockage of the concerned enzyme in this study? Oxidative deamination, oxidative decarboxylation, oxidative phosphorylation, substrate level dephosphorylation, substrate level phosphorylation. MCQ 3. Which of the following enzymes catalyzes the phosphorylation of a substrate with the use of inorganic phosphate? Glucose 6-phosphate phosphatase, glyceraldehyde 3-phosphate dehydrogenase, phosphofructokinase, pyruvate dehydrogenase complex, alpha-ketoglutarate dehydrogenase complex. MCQ 4. A 6-year-old boy has been prescribed fluoride-containing toothpaste for dental caries. Which of the following enzymes of glycolysis is inhibited by the particular content of prescribed toothpaste? Aldolase, enolase, glucokinase, hexokinase, phosphotriose isomerase. MCQ 5. Which of the following enzymes is responsible to convert glucose 6-phosphate to fructose 6-phosphate? Kinase, oxidase, isomerase, dehydrogenase, hydrolase. Please write answers in the comments or contact drmjot@yahoo.com. End of Part 1 of 3 of Chapter 10. Thank you.